Good evening, everyone. Welcome to the week 6 live interaction session for the course Waste to Energy Conversion. So, as usual, let's just wait for a few minutes for others to join before we begin our session for today. So, let's start our session for today. So, let me just share my screen. And yes. So, I hope my presentation is visible and I'm audible. So, welcome everyone to the week 6 live interaction session for the course Waste to Energy Conversion. This course is offered by Professor Prasenjit Mondal from the Department of Chemical Engineering, IIT Roorkee, under the NPTEL SWAYAM program. Myself, Pratyumnan, a PhD research scholar from the Department of Chemical Engineering, IIT Guwahati. I am responsible for conducting the live interaction session for this course, Waste to Energy Conversion, in the Jan May 2026 semester. So far we have traveled almost five live interaction sessions are over, and today we will complete the sixth one, which makes up to 75% of the course. So till the last live interaction session was what we mainly focused on the thermal conversion route for the extraction of energy from the waste material. We dealt with the three different types of thermal conversion routes. The first one is the incineration, also called as combustion, then gasification and pyrolysis. Over there, as in short to speak about, we discussed on what is the fundamental difference between the three different techniques and what is the composition of the products that we can extract from them. So I hope everybody is able to recollect. So till week 4 live interaction session, our main goal was to understand the fundamentals of these three different processes and what are the types of reactors that we can use in order to extract energy from this waste material. The last live interaction session was basically more on improving the energy efficiency process in the thermal power plants. So we discussed on the various factors that typically affects the energy generation in a power plant, and we also discussed on the ways to combat them. After that, we discussed about a different concept on energy production from the plastic waste. So as I described in the last live interaction session, also in the course, the plastic waste are getting generated at an exponential rate. So human life without the use of plastic has is somewhat unimaginable. We have almost traveled to that particular extent. So using plastic leads to generation of plastic waste, and these plastic waste often ends up in this environment, specifically in the oceans. So when it enters inside the water bodies, due to the ocean currents, wave currents, the plastic waste tends to break down into smaller particles. They do not degrade, but they do not degrade into you can say environmental compatible materials, but then in, they break down and remain in the ocean, which is then taken by the aquatic animals, and humans eat these aquatic animal, and then the microplastic enters inside our body. So there are several literature reports where people have identified the presence of microplastics in human body in multiple different organs. So yes, that is a whole big area of studies where they are trying to discover how to minimize the microplastic waste, how it enters inside the human body, and where does it goes and gets deposited at a larger concentration, and so on. So that's entire field of research that is actually going on in that field, and recently many concerns have arisen in the field of microplastics. So after that, we discussed about the gas cleanup. So the gas that gets generated, the biogas or the syngas that we obtain from these kind of energy waste to energy conversion process, once their use is over, either in the form of either to run a steam turbine or to a gas turbine, these gases needs to be let out in the atmosphere. But before we do that, we need to do some pre-processing so that they do not pollute the environment or the atmosphere. So for that, we discussed about multiple different types of gas cleanup equipments. The four main things that we discussed was on electrostatic precipitator, which is commonly used in the industrial scale. Then we discussed about gravity settlers, fabric filters, and cyclone. So over there, specifically on ESP, we discussed on the working principle. How does the ESP work? So in short, to speak about very high DC voltages given to the electrodes, the parallel plate electrodes, which generate a very high electric field, and when the gas pass through this electric field, the gas becomes negatively charged. The electrode plates are usually positively charged, so the particulate matter in that gas are negatively charged, so they are attracted towards these positive plates, and then they get deposited on the plates due to attraction. So when that typically happens over and over a period of time, this particulate matter keeps getting accumulated on the surface of the electrode and covers the electrode surface. Once a particular limit is reached, these electrodes are disconnected from the DC voltage supply, and then the layer of the particulate matter that is deposited is cleaned up before these electrode plates can be used for the next cycle of purification, purification of the gas, so that it can be let out in the atmosphere. So usually these norms of what should be the particulate matter, their types and size is usually defined by the Central Pollution Control Board, CPCB. So they have a definite norms on what should be the particulate matter and its size. So if anyone is interested, then you are welcome to look it in look online so that you can get what are the metrics that they follow. So today we move our focus to a completely different types of waste, that is biological waste. So today we will discuss more about the energy production from the anaerobic digestion, and after that we will discuss about something so called as microbial fuel cells or called MFCs. So if you have to distinguish these conversion routes, previously we discussed about the thermal conversion routes. So you can see in the left hand side that the biomass material is usually burnt and the energy is released, and that energy is what is getting used up, and for that we use three different processes. Whereas if you see in case of the biological routes for the conversion of waste to energy, we tend to convert the biomass to biogas, specifically in the form of methane and carbon dioxide. And how do we do this process biologically? For that, we go for something so called as anaerobic digestion. So, anyone, any idea on what is the word, what does this digestion process actually means? So, here, in case of biological conversion, we convert the biomass to biogas, and for that we use a process so called as anaerobic digestion. Now what is this anaerobic digestion, or what happens in this anaerobic digestion process? Anyone, what is anaerobic digestion process? So anaerobic digestion, yeah, okay, somebody, yeah, it's the organic matter, the you can say that the microbes actually degrade the organic matter in absence of oxygen. So that's what I was looking for, that we do this entire process in absence of oxygen. So in case of anaerobic, it is absence of oxygen, and in case of aerobic, it is in the presence of oxygen. So the entire process, what you do, if you carry out in the presence of oxygen, it's called aerobic. Absence of oxygen, it's called anaerobic. So so you do this biomass to biogas conversion, and then whatever the residue that is left out after the digestion process, that is that residue is often used as a compost or fertilizers compost. So that compost is used as a fertilizer for the plant. Then you also get hydrogen gas as well as reusable water. So there is one main you can say difference between this thermal conversion routes and biological conversion routes. One particular difference is in the amount of solid content that you can digest through these two processes. So if you see in case of biological conversion routes, the solid content needs to be only 8%, and the rest 92 percentage is typically water. So whereas in case of the thermal conversion routes, if you see, it is not as in that case you have more amount of solids, but then the moisture is typically very less. The major reason being is that if you have moisture, it typically decreases the energy conversion process, or you can say the energy efficiency gets reduced. However, in case of the biological conversion routes, what we need to take care is that the solid contents needs to be only to up to 8 percentage, and then the rest of the uh, raw material, or you can say the feed, is usually water. So this is one of the main, or you can say a very important mm, kind of difference between this thermal conversion as well as that of the and the biological conversion routes. So this biogas that you typically obtain, it usually has methane in major quantities, and apart from that carbon dioxide is also generated. The rest of the gas that gets generated, like nitrogen gas, hydrogen gas, and oxygen, and and hydrogen disulfide, sorry, dihydrogen sulfide, are usually minor quantities. They are released, but then their percentage in the biogas remains very less. So apart from this biomass, biomass to biogas conversion, you can also convert this biomass to other kind of biofuels like alcohol production, and that typically is what you called as ethanol fermentation process, where you break glucose to ethanol, and carbon dioxide is also released along with this. So if you see this anaerobic digestion process, it typically happens through multiple different stages. It's not that the biomass, when you just when you just do some micro when you just add microbes to this biomass, and then if you allow the entire process to happen in the absence of oxygen, you will directly get biogas. So it's a chain of process that happens in this anaerobic digestion process. So anyone who knows what are the list of process that is involved in this anaerobic digestion process? What are the steps involved in the anaerobic digestion process? There are four different steps that is involved in the anaerobic digestion process. Yes, anyone? Yes. So it is, the first process is hydrolysis. So hydrolysis is nothing but here. So usually when you say biomass, they are typically in the form of starch, protein, and fat. So they are all very long chain of molecules. So that complex compound are usually broken down into simpler organic compounds. So convert conversion of complex compounds into simpler organic compounds. So if you, for example, if you take starch, it is usually broken down in the form of simple sugars like glucose, or yeah, so the glucose is the simplest sugar. So glucose or fructose or sucrose, those kind of simple sugars. And if you take it in the form of proteins, you have amino acids. So proteins are usually broken down into amino acids because amino acids are the ones that actually form proteins. So their basic entity of protein is amino acids. And if you go for fats, then you will have shorter chain of fatty acids, or you can also call them as peptides. So hydrolysis is typically a process where you tend to break the complex compounds into simpler organic compounds. So that simple why? So that their digestion, so that the digestion process can be very easy. It takes lot of effort for this bacteria to digest on a complex compound, right? So if their molecular structure is broken down, then the digestion process will be far more easy. So that is the first process of digestion, is breaking complex compound into simpler organic compounds. Second thing is the acidogenesis. So as the name suggests, the acid generating bacteria, or the you can say acid generating bacteria usually feed on this simpler organic compounds to generate short chains of fatty acids. So acid generating bacteria breaks, you can say that the acid generating bacteria, they produce acid, and that acid acts on the simple organic compounds what we have obtained from the hydrolysis process. So the second process is called acidogenesis. Then there is a third process which is called as acetogenesis. So acetogenesis is a process where acetic acid is generated. So in case of acidogenesis process, the acid usually acts on the simple organic compounds, and in case of acetogenesis process, what happens is that whatever product that you get from this acidogenesis process is further processed so that acetic acid gets generated from the system. So this process is called acetogenesis, where acetic acid production starts or happens. Then there is another one process that is called methanogenesis. So here typically the methane is produced. So from the acetic acid, the bacteria acts on this acetic acid to convert them into methane plus carbon dioxide. So the methane is generated in this process or in this step. So that step is called as methanogenesis. So typically the reaction is something like this. So this is the methanogenesis process. So there are totally four steps that is involved in the anaerobic digestion process. One is the hydrolysis, second one is acidogenesis, third one is acetogenesis, and fourth one is methanogenesis. So that's how you basically go from biomass to biogas conversion process. So just a second. So now let's come to this ethanol fermentation process. So this is how you typically do this anaerobic digestion process, as we discussed previously, where you basically go from or convert this biomass into biogas, and for that you go through four different processes. The first process being hydrolysis, followed by acidogenesis. After that you have acetogenesis and then methanogenesis. Now we come to fermentation process, where you get ethanol from this kind of simple sugars like glucose. So C6H12O6 typically represent for glucose. So this glucose, if you do fermentation process, you get ethanol and carbon dioxide. So in order to do this fermentation process, you usually use yeast. So yeast is commonly used for the ethanol fermentation process. So as such, just like anaerobic digestion, this fermentation process also has a series of steps in order to obtain ethanol from this simple glucose that you get from the simple glucose. So first you basically go for pre-processing steps wherein the impurities are removed. Then the fermentation process actually happens, where this organic matter, or you can say that glucose, is further broken down to produce acid and simple sugar, and then to basically produce ethanol. So in the fermentation process, this entire breaking down of the glucose into simple sugars as well as that of the ethanol happens. Then you go for the distillation process, where you concentrate that ethanol so that you can get high concentration ethanol from the fermented broth, you can say. So once that ethanol is concentrated, then you go for a dehydration process, where you tend to obtain anhydrous ethanol. So the major reason why, so ethanol, the major reason why we try to produce this is basically because of its calorific value. So ethanol can be used as a biofuels. So that's what we have written: conversion of biomass to other biofuels, alcohol production. So the ethanol that we generate, it, apart from the commercial purpose, other commercial purposes like used in laboratory and laboratory as chemicals, this ethanol can also be used as a biofuel because of its higher calorific value. Of course, in order to use ethanol as a biofuel, you need to actually blend it with gasoline, so that their energy can be enhanced, the calorific value can be enhanced, so that they become apt to use it as a biofuel for any kind of vehicular applications. As such, as pure ethanol is often not used as a biofuel, typically being is that their calorific value do not actually match up with the calorific value of the targeted applications. And there arises a question now: if gasoline is already there, and if we have to if gasoline is already going to be present, and in order to use ethanol as a biofuel, I need to actually mix it with the gasoline so that they can be used as a biofuel as such for my application. Then why should I actually go for ethanol itself as a fuel? Why can't I just stick to gasoline? So I hope the question is clear. So gasoline is typically being used. Now I am going to give you as ethanol. as a biofuel for your application, but its calorific value is low. So I have to blend that ethanol with the gasoline so that I can use it up for my application. Now if this is the case, then why I need ethanol itself in the first place? Like, why ethanol, why there is a promotion for using ethanol as a biofuel? Octane rating. Okay, yes. So uh, ethanol as such is having lesser octane number. So for that we actually blend it with gasoline. One of the main reason is to basically make sure that the process is environmental friendly. Actually, if you use gasoline, the amount of vehicular emissions that you are going to obtain will be much greater compared to using ethanol plus gasoline mixture. So in order to reduce the vehicular emission and make the process more environment friendly, we go for using ethanol as a biofuel. So here as such we have to remember few things. So in case of ethanol fermentation process, we usually try to extract ethanol from glucose using fermentation process with the help of yeast. Now the entire fermentation process can be divided into series of steps, just like in case of anaerobic digestion. So first process is to do, first is a pre-processing step where you try to remove the impurities from the feed. Then you go for the actual fermentation process where the ethanol is produced. The glucose is broken down into simple sugars, further simple sugars, which is then converted to ethanol. That ethanol undergoes distillation, because that ethanol that is produced through the fermentation process will not be pure. It will actually it will be actually a mixture. So that, so to extract pure ethanol from that, you actually go for the distillation process. So ethanol gets concentrated, concentration of ethanol happens. Once distillation process is over, you basically go for the dehydration step in order to get the anhydrous ethanol. So here, in order to use ethanol as a biofuel, we go for anhydrous ethanol. Again, the same reason, if there is going to be any moisture, that will affect the calorific value. So this ethanol cannot be as such used in case of as a biofuel because it carries less energy. So often it is blended with the gasoline. So now, as we discussed just before, if I have to blend it as such with some other already widely used fuel, then why I need to use why I need to use ethanol itself in the first place? The main reason being is that the vehicular emission. So from gasoline, the vehicular emission is more, that causes environmental pollution. If we try to basically mix both of gasoline as well as ethanol, then we can reduce or cut down the vehicular emission. That makes the entire process more eco-friendly or environment friendly. So this is the place of end of discussion of the anaerobic digestion and ethanol and the fermentation process. So next we will actually discuss about the microbial fuel cell. So before moving into that, any questions so far on what we have discussed till now? Anyone? Okay, so I think we can move on for microbial fuel cells. So, microbial fuel cell, as such, I mean, before starting with the concept of microbial fuel cell, how many of you actually here know what's a fuel cell? Actually, fuel cell is more of a energy generation device, or you can say energy is produced there. So, in case, so a fuel cell is more of an electrochemical device. So here what typically happens is, so before discussing about microbial fuel cell, let's just think about or talk about fuel cell as a individual concept. Then we will discuss about this microbial fuel cell. So in fuel cells you have multiple different types. Among them, microbial fuel cell is a one. So fuel cell is actually a type of an electrochemical cell. So any particular electrochemical device, or you can say any particular cell like batteries, or you can say fuel cells or supercapacitors or any kind of a cell, you usually classify them into two different types. So one is an electrochemical cell and another one is an electrolytic cell. So let me just write that here. So usually we divide them into a electrolytic cell. In electrochemistry, any particular system in electrochemistry, we try to classify them as an I, uh, we try to classify them either as an electrolytic or as an electrochemical cell. So both of these cells usually use or usually operate via different mechanism. In case of electrolytic cell, what actually happens is electrical energy drives chemical reactions, or you can say electrical energy is converted to chemical energy. Whereas in case of an electrochemical cell, chemical energy is converted to electrical energy, or the chemical reaction generates electrical energy. So the it's just that they are opposite of each other, but then these two different mechanisms have wide variety of applications. So the main thing that we need to remember is the operating principle. So this is their operating principle and the main difference between both of them as well. In case of an electrolytic cell, electrical energy is converted to chemical energy, whereas in case of an electrochemical cell, chemical energy is converted to electrical energy. So depending upon the target application or where we want to use this cell, we choose electrolytic or electrochemical cells. So fuel cell, usually what happens in case of a fuel cell is that, or you can say a hydrogen fuel cell is that the hydrogen gas is passed through the anode. So usually a fuel cell, the construction is somewhat similar to what is there in this picture. So for any fuel cell or an electrochemical cell, if you take, you will have an anode, cathode, and then both this anode and the cathode will be separated by a membrane. The anode will be surrounded by a electrolyte, as well as cathode will also be having an electrolyte. So in case of a fuel cell, what actually happens is the anode gets hydrogen gas. So hydrogen gas is usually pumped through the anodic chamber. So this hydrogen gas comes in contact with the anode electrode, and it basically generates H plus ions, plus it undergoes oxidation to generate H plus ions plus electrons. So let me just write the reaction. So let me first discuss about the basic fuel cell, that is the hydrogen fuel cell. Then let's come to the concept of microbial fuel cell. So in anode, the hydrogen gas is pumped inside the anode. So this hydrogen gas comes in contact with the anode electrode, and then it undergoes oxidation, where hydrogen gas gets converted to H plus ions, and that releases along with the release of two electrons. Similarly, in the cathode side, you pump or you feed oxygen. So oxygen is fed to the cathodic chamber or to the cathode chamber. This oxygen comes in contact with the cathode electrode. Now here in the cathode, reduction of the oxygen takes place. So what happens is these H plus ions and the electrons that are generated in the anode moves from the anode to the cathode chamber. So the H plus ions move through the membrane, and then the electrons move through the wire. So you have a connection between the cathode and the anode. So the electrons moves through the wire, whereas the H plus ions move through the membrane. So both the H plus ion and the electron meets the oxygen at the cathode and undergo reduction. So it is reduced to 2OH minus. So this is what happens in cathode and anode. So together, if you see, have I written it? Yes. So the overall, what happens is, overall reaction is H2. I haven't balanced this, so I don't think, I mean, I hope you guys can balance this. Instead of this, I'll write H2 also. This will be better and easy to understand. So the H2 gas, so the oxygen reacts with the H plus ions plus electrons, leading to the formation of H2O. So the water is generated through the fuel cell or from the fuel cell. This electron basically is responsible for the generation of energy. So as such, from a hydrogen fuel cell, you can actually get clean water. The water that gets produced through this process is extremely very pure form of water. And the second thing is, you can extract energy from this process. So here we discuss, so here a chemical reaction is giving energy, or is basically generating energy. So hence, fuel cell is a type of electrochemical cell, or these kind of electrochemical cell where the chemical energy is converted to electrical energy, is also called as a galvanic cell. So fuel cell is a electrochemical cell, or you can also call them as a galvanic cell. So this is the concept of hydrogen fuel cells. Any questions? And the amount of voltage that you can obtain from this hydrogen fuel cell, theoretically it is 1.23 volt, which is also the thermodynamic potential for water splitting. But since, but in practice, you cannot obtain 1.23 voltage. Generally the voltage that you can generate from this process floats at around zero point 0.8 to zero point 0.9 volt. So yeah, so that is one more thing. So any questions so far related to fuel cells, hydrogen fuel cells? So because same concept we will use it in microbial fuel cell. Over there, instead of hydrogen gas, we use organic matter and microbes. Any questions? Oh, okay. Any questions related to hydrogen fuel cells? So next we will now discuss about microbial fuels. So as I said, same concept in is also used in case of microbial fuel cell. The only difference being is that in case of your anode, instead of using hydrogen gas, or instead of pumping in hydrogen gas in the anodic chamber, we will use organic matter and microbes to generate the electrons. So the typical reaction, just write anode is CXHY. So that typically is your organic matter. So any kind of organic matter it could be this, and also it has water. So this organic so microbes are also present. So the microbes feed on this organic matter in the anode compartment to generate CO2 plus H plus electrons. Now same way the electrons moves from the cathode to the anode, from the anode to the cathode through the wire, and then the H plus ions move from the anode to the cathode via the membrane. So in the cathode section you have oxygen, and this H plus ions as well as the electrons meet to form H2O. So if you try to balance this reaction, it should be 4H plus 4 electrons gives 2H2O. So this is the reaction that happens in microbial fuel cell. So here the CO2 actually gets generated in the anode compartment, which is usually collected time to time from the anodic chamber. Again, as we discuss, as I said, even in case of the hydrogen fuel cell, even though the energy or the voltage that you can obtain from this hydrogen fuel cell is 1.23 volt, however, getting that particular voltage is often difficult. The major reason being is that there are several losses that happens within the fuel cell. So let's look at the types of losses. So there are multiple different types of losses, like concentration losses, activation losses, ohmic losses, then losses because of the loss in the H plus ions while during the movement, during their movement through the membrane, then the electron losing the energy, that is what is ohmic loss. So all these contributes to the decrease in the energy generation from the fuel cell. So similarly, in case of microbial fuel cell, all these problem remains. So one thing that you have to look up here is, so all these things remain. So our major motive is to basically make sure that the electron transfer is more rapid, more amount of electron transfer happens, so that more amount of energy can be generated from the system. But if more amount of the electrons has to be produced, there is a catch here. So here what happens is, in case of anode, the hydrogen gas, when it comes in contact with the anode electrode, this hydrogen forms H plus plus forms protons and electrons. But in case of microbial fuel cell, there comes a question on how does this microbe acts on the organic matter. So CO2 generation is, we can understand that how the CO2 may get generated, but how does this electrons get generated by these microbes? And if so, the electrons are generated, how does this uh, how does this electrons actually move to the electrode, and from the electrode it actually goes to the, move to the anode electrode, and how does it move from the anode electrode to the cathode? So there lies a question. So in case of microbes itself, in the microbial fuel cell, there are different types of microbes which you can actually use. Some of them are called as exoelectrogens, where they digest this organic matter. The microbes digest this organic matter, but then the digestion happens outside of its body. From there, the electrons that gets generated goes to the anode electrode, which then moves to the cathode electrode. So let's say, for example, in case of an exoelectrogens, where the electrons are, where the digestion process happens outside and the electrons are generated, now there comes a question on how these electrons that gets generated actually moves to the anode. Is there any kind of a particular mechanism, or does it just go just like that, it gets attracted to the anode, and from the anode it moves to the cathode? So That typically depends upon the type of microbes that you use, and in order to make sure that this electron transfer from the waste matter to the electrode is more efficient, for that we use some sort of a mediators. So those mediators help the electron to move from this organic waste to the anode electrode. So there are different types, varieties of mediators or external agents are used to make sure that this electron transfer is more or happens more conducively or easily. So their types is, one is thionine, second one is methylene blue, and then you have methyl biologen, and then you can also use humic acid. So their main role is to promote the movement of the electrons from the organic waste to that of the anode electrode. So as I said, when more amount of electron transfer happens, energy generation process will be more and you can obtain more voltage. Of course the losses will still remain, but the loss due to this electron transfer will be greatly reduced using this external agents. So again, there are multiple different factors that affects your MFC process. The type of anode cathode and then the membrane that you use also affect the energy efficiency of the process greatly. So for example, if you use, let's say in case of the membrane, you typically use Nafion membranes. So if you use um, the Nafion membrane as such is used for hydrogen fuel cells, but let's take an example of that Nafion membrane itself. So if you use a thicker membrane, what happens is it becomes difficult for the H plus ions to move from the anode to the cathode. So that decreases the efficiency of the process, because the oxygen that is at the cathode electrode will be devoid of the H plus ions, and because of that it makes the uh, energy generation as well as the generation of water difficult. But if you make the membrane too thin, then what actually happens is the anode and the cathode compartment may also mix up with one another. There won't be much of a physical separation between them, and mixing of both these of the contents in both this chamber is also possible. So this is one way on how the membrane thickness affects the process. Again, membrane apart from the thickness, it's also the type of membranes, the molecular structure of the membrane also greatly influence the process. The type of catalyst also have a greater impact on the energy generation process. Whether you are going to use PT, I mean PT in the sense platinum or platinum supported carbon or polyaniline or MnO2, each of these catalyst will vary the fuel cell, the microbial fuel cell process and the energy that you can obtain from them. So if you talk indeed about MFC process, you can see a big list: temperature, organic matter, what type of organic matter you use. I mean, is it very complex organic matter that it becomes difficult for the microbes to digest, or is it in the form of simple sugars, and at what temperature you do this process. Electrode substrate that you use, whether if it is graphite or any other kind of electrodes. So the conductivity of the electrode basically becomes an important factor while transporting the electron from anode to cathode. If the conductivity is low, electron transfer is hampered, which reduces the energy generation from the fuel cell. pH of the electrolyte, types of catalysts, types of microorganism, whether they are able, as I said, exoelectrogens and endoelectrogens, how the digestion of the organic matter happens in these microbes affects the process. Type of oxidant in the anodic chamber affects the process. So all these process affects the energy that you can harvest or generate from the microbial fuel cell. So microbial fuel cell as such is an interesting concept. A lot of research is going on in this field related to fuel cell as well as microbial fuel cell. So again, compared to hydrogen fuel cell, the amount of energy that you can generate from the microbial fuel cell remains low. So that becomes a interesting research gap or a research direction, which basically motivates many researchers to work on this microbial fuel cell. And again, it also, since you obtain energy from the waste, it's also a more sustainable waste management approach, or you can say sustainable energy generation approach, because in case of a hydrogen fuel cell you need to pump hydrogen gas. So again, hydrogen gas has its own limitations, such as it is flammable, and then it has um, like availability, and it is flammable. That is the main aspect of the main what do you say, limitations of that. Whereas in case of a microbial fuel cell, you avoid that hydrogen gas feeding into the anodic chamber, feeding to the anodic chamber. So my this thing is a lot of research is happening. So it's like a very encouraging field. So many of you, if you are interested, you can have a look at. There are several research papers already out there in the field, each explaining how uh, a particular parameter or a process is optimized and how that affects the energy generation of the system. So it's a great area of research to focus on. So, as far as the course is concerned, or the week 6 live interaction course content is concerned, it ends here. So, after this, we will be solving as usual. We will be solving the problem. So, if you have any doubts on what we have discussed so far, please feel free to ask before we move towards our what do you say, assignment problems. Any questions? Again, apart from hydrogen fuel cells and microbial fuel cells are just two different examples that I have given. There is a whole big class of different types of fuel. This thing, what do you say? Fuel cell. There's something called methanol fuel cell. Again, each of them have their own operating range. So, hydrogen fuel cells, as far as I know, it usually operates at 25 to, I mean, in the ambient conditions, and it can go a maximum up to 50 to 60 degrees Celsius. So, the major reason being is that uh, beyond 50 to 60 degrees Celsius, that the Nafion membrane that we just a second that we use here, that Nafion membrane is usually in its hydrated form. So that only so that the hydrated Nafion is what it actually helps the H plus ions to move from anode to cathode. So when you actually increase the temperature to 50 or beyond 50, let's say 60 or 70 degrees Celsius, what happens is that the hydration level of that membrane decreases, and that creates an issue for the movement of the H plus ions. And one more reason is that H2 gas that you use, as I said, it's flammable in nature. So a high temperature also increases the risk that there can be a fire hazard. So mainly due to these two reasons, hydrogen fuel cells is often cannot be used for high temperature applications. But again, as such, there are many different types of fuel cells which can be used even for high temperature applications, and it's a promising area of research because people are trying to integrate this fuel cell into vehicles to form e-vehicles or electric vehicles. Again, there's a long way, a long road to actually move forward, but still, it's a promising direction. So if you have no questions, let me just check once. Okay, if in case if you have no questions, I think we can move towards problems. So we have totally six types of MCQ questions, and then again we'll move towards the big problems. I mean the numerical problems. So the first question is, which of the following is a metabolic process that converts sugar to acids, gases, or alcohol? You have anaerobic digestion, pyrolysis, fermentation, and gasification. Which of the following metabolic process converts sugar to acids, gases, or alcohols? Is it anaerobic digestion, pyrolysis, gasification, or fermentation? A. Anaerobic digestion. Okay. But what about fermentation? Pyrolysis and gasification may not be the answer because there you do not convert the sugars to acids or alcohol. There you take the biomass and then you treat them under different temperature conditions and oxygen conditions or oxygen conditions, and then you obtain gases and solid char in case of pyrolysis. So what about fermentation? So question says sugars to acid, gases, or alcohols. So keep that in mind, and then mm, so based on that, what could be the answer? So question says it is convert sugar to acids, gases, and alcohol. So in case of anaerobic digestion, we take biomass and then that biomass undergoes hydrolysis, acetogenesis, and methanogenesis. You don't try to, you never get alcohols from it. So whereas in case of the fermentation process, we take sugars like um, for example, sugars as such, you take glucose or fructose or sucrose and then break them down and then convert that sugars into acids or alcohols, and then that alcohol is what you use it as a biofuel. So The correct answer is fermentation is a metabolic process that converts acid gases that converts sugars to acid gases or alcohol. Any ambiguity related to the answer for the first question? Any confusion? I hope so. No. So if not, then let's progress to the next one. What are the factors that affect the yield of biogas? So the biogas that gets generated through the anaerobic digestion process. So that process typically happens in an anaerobic digester. So apart from I mean other so as such, this conversion process of biomass to biogas is usually dependent on several different parameters. So which of the following parameters that is here tends to affect the biogas yield? Is it the dry matter, temperature, pH, or all of these? Just a second. So sorry. So I had to change the network connection. So let me just share. Yes. So the question is, what are the factors that affects the biogas yield? Anyone? Is it dry matter, temperature, or pH, or all the three? Yes, so all the three affects the yield of biogas. Of course, it's not just only these three parameters. There are several other parameters that affects the yield of biogas. So anyone apart from these three, any other yield, sorry, any other factor that affects the yield of biogas? Any one, just one or two factors other than the one that is mentioned in the options? Any other factors? So kindly have a okay, yeah. So retention time, yes, that is one factor which affects. So I'll just mention it here. Retention time. So again, there are multiple other factors. So please have a look at it in the course lectures. So each of these factors have a particular effect over the yield of biogas. So all the parameters need to be optimized so that we can maximize the yield of biogas. Any particular factor which is either present lower or higher than what it is actually required or the optimized amount will positively or will negatively affect the yield of biogas. So these are the three parameters that is mentioned here. There are several others, so kindly have a look at it and how they affect the yield of biogas from the course lectures. So next question is electrochemical cells. So this is just what we discussed a while ago. So electrochemical cell converts what type of energy into what other form of energy? Is it chemical to electrical, electrical to chemical, chemical to mechanical, mechanical to chemical? E- Chemical energy to electrical energy. So electrochemical cells, also called as galvanic cells, they convert chemical energy into electrical energy. So in case of the electrolytic cell, it is the opposite. Electrical energy is converted to chemical energy. So let's move on towards the next question. I think other than the first question, the second two questions are simple. So I hope there won't be much questions there. Anyway, let's go see the fourth question. Identify A, B, and C in the fuel cell. So you have the fuel cell architecture here. You are supposed to let me know what is A, B, and C. I mean the components. Is A anode, B cathode, and C the proton exchange membrane? Or is A is cathode, B is anode, and C is the proton exchange membrane? Or A is anode, B is cathode, but C is a rigid wall? And D is A is cathode, B is anode, and C is a rigid wall. Here, the distinguishing factor between anode and cathode will be based on the type of gas, or you can say the reactant that is used. So it's so yes, and also you can say that from the flow of electrons. A. A is anode, B is cathode, and C is proton exchange membrane. So the basic idea for the answer is that A is having hydrogen gas and B is having oxygen or air. So that typically means in case of a proton exchange membrane or hydrogen fuel cell, A is anode, B is cathode. Or you can also distinguish this from the arrow marks. So if you see the electron, it is moving from the left side to the right side. So left side should be the anode and right side should be the cathode. Similarly, if you observe the arrows or direction of the H plus ion, it is moving from the left hand side to the right hand side. So left should be anode and the right should be cathode. And through a rigid wall, H plus ions cannot move. So it should be a membrane, that is the proton exchange membrane. So option A is correct. A is anode, B is cathode, and C is proton exchange membrane. So I hope no doubts. If you have any doubts, please let me know, because I won't be doing the question 5 and 6 because I hope all of you know the answer for the question already. So any doubts related to the 4 questions that we discussed? Anyone? Mm, okay, so that is the okay. So I hope no one has any doubts. So if that is the case, then I will move forward with the numerical problems. So in case if anyone has any doubts, then you're welcome to let me know, or otherwise. So let us start solving the problems for today's live interaction session. So today's problem solve the numerical problems that we have is somewhat like we'll be having a mix of week 4, week 5, and week 6. So these were some of the questions that were like that went as a backlog in the previous sessions due to time constraint. So we will try to finish those backlogs today. So let's first start with this question before we move towards the before previous one. This is quite easy. So it's about the densification of solids where you try to produce briquettes. So a 10-ton hydraulic jack was used to lift the machine components and compress the briquettes. The compaction force can be calculated using the pressure. The pressure read from the pressure gauge connected to the hydraulic jack is 17.5 kilonewton per meter square. The machine has 50 moles and the diameter of the briquette sample is 30 mm. Calculate the compaction force. This question is simple, so let's start with this. Anyone, any idea on how to proceed steps? So what is, how do you calculate force you from pressure? Like, yeah, the question actually gives a hint. The compaction force can be calculated using the pressure. So what is, how do you relate force and pressure? The, the formula for relating force and pressure. Um, So can we write force is equal to pressure times area? Yeah. So pressure is equal to force by area, or you can write force is equal to pressure in pressure multiplied by area. So pressure is already given, that is 17.5 kilonewton. If anyone has solved already, if you have the answer, then please let me know. Let, and we need to calculate area. So area of the mold we need to calculate. So that will be pi by 4 into diameter of the briquette sample whole square. So area of mold equals to pi by 4. So that is 3.14 value of pi times, I am going to convert millimeter to meter, so that is 30 by 1000. The whole square. So these many meters square for 1 mole. Now he is saying the machine has 50 moles. So the area of 1 mole times the number of moles. So that means it will be 3.14 multiplied by 0.03 whole square multiplied by 50. 50 times 3.14 times 0.03 whole square. So this is the area of the machine. Now we have pressure, we know the area. So the compaction force is nothing but pressure times area. So that will be equal to 17.5 times 50 times 3.14 times 0.03 whole square. Now if you calculate this, you will get force in terms of kilonewtons, because pressure is 17.5 kilonewton per meter square. So this will be in terms of kilonewtons. So what would be the answer? So let me just use calculator. So 17.5 times 3.14 times 0.50 times 0.03 times 0.03. This entire divided by 4. I forgot to mention that there. So that would be 0.618 kilonewton, or if you want to write it in terms of Newton, then it would be 618.18 Newton. So the compaction force is 0.618 kilonewton or 618.18 Newton. So I hope not there won't be much doubt related to this. If you have anything, you please feel free to ask, or else we can move forward to the next question. Anyone, any doubts this in this question? Yes, the answer is 0.618 kilonewton. Correct. So that's what we are also getting. So yeah, thanks for sharing the answer. So anyone with any other doubts or with any doubts? So if you have no questions, then let's move to the next question. So in a just a second before that. Okay. In a laboratory, 100 gram of waste plastic sample is pyrolyzed in a batch reactor which weighs 900 grams. The weight of the collected oil is 60 grams and the final weight of the reactor is 925 grams. Calculate the yield of gas, liquid and solid fractions. So you have 100 gram of waste plastic sample. Now you need to pyrolyze that so that you can obtain energy from that. And that you actually do it using a batch reactor. That batch reactor weighs around 900 grams. Weighs exactly 900 grams. Weight of the collected oil after the process, whatever the oil that you are collecting, is 60 grams and the final weight of the reactor is 925 grams. Calculate the yield of gas, liquid and solid fractions. Any answers? This question is a very simple one. So, so you have 100 grams of plastic waste and you have oil that comes out, the weight of oil is 60 grams. So the yield will be nothing but 60 by 100. So that is nothing but zero point 0.6. And weight of the reactor before and after is 925 and before is 900. So that would be the weight of the solid fractions that is getting generated. That is the reason the batch reactor's weight has got increased. So weight of solids is final weight minus initial weight. So that would be 925 minus 900. So that would be 25. 25 grams of solid is actually uh, generated in the process. So its yield will be 0.25. 25 by 100. So that would be 0.25. Now based on this, calculate the yield of the gas. Yield of the gas. So So nothing is known about the yield of the, I mean the weight of the gas that gets generated or anything. So what we can do is we know that since the the yield, it's not, since we do not have any data on the fact that whether entire plastic waste sample is going to be converted to gas, liquid or solid, or is it only a small fraction of them, or does it have a conversion efficiency, conversion percentage? So since we do not have any data on that, we can assume that all the plastic waste sample is going to get converted either as either it is going to get converted to gas, liquid or solid. So with this assumption, what we can do is to calculate the yield of the gas. It will be nothing but 100 minus 60 minus 25 by 100. So that would be 0.15. 15. So 15 grams. So yield will be 0.15. So yield of oil is zero point 0.6. Yield of gas is 0.15 and yield of oil, solid is 0.25. So any questions related to this? So if you have no questions, we can move forward. We have approximately 10 minutes of time with us, and I think we can finish one more question. So if you have doubts, feel free to ask me now, or otherwise we can move towards the next one. Any questions? So let's move towards a venturi scrubber problem. So a venturi scrubber is used to collect the PM, that is your particulate matter, from a gas stream. The liquid flow rate through the scrubber is 45 gallon per minute per 100 cubic feet per minute of gas, and the relative gas velocity to liquid is 200 meter per second. The gas is air at STP and carries particles of density 800 kg per meter cube. Determine the efficiency of the scrubber as a function of the particle diameter. Consider the viscosity of the gas as 1.8 times 10 to the power minus 5 kg per meter second, and the empirical factor for the throat geometry and other parameter K is 0.25 as 0.25. So this is a question related to gas cleanup on venturi scrubber. So we are given few data, and based on that data we are asked to calculate the efficiency of the scrubber as a function of particle diameter. So it's going to be a expression or equation, you can say. So take a few minutes to think on how to solve before moving on towards the solution. So what's the formula for calculating the efficiency of a venturi scrubber? Sorry. So the formula for calculating the efficiency is nothing but it is 1 minus exponential of minus KL times pi, the whole power 0.5. This is the formula for calculating the efficiency of the venturi scrubber. Now we need to, there are expressions for the phi as well as the diameter of the particle. So let me just write. C times rho of the particle, rho of the particle times dp square, so that is diameter of the particle, times Vr by 18 dO times mu g is nothing but the viscosity, is the viscosity. Mu g is the viscosity uh, of the gas. So based on that, we will be able to calculate the expression for phi, and dO is, can be related as 16,400 by Vr plus 1.45 L to the power 1.5. Now these are the expressions. We know Vr is nothing but it is 200 meter per second. So if you substitute this here, and we know L is nothing but 45. So if we substitute here, D naught will be nothing but 16,400 by 200 plus 1.45 times 45 whole power 1.5. So that would be, what would be 16,400 by 200? 82 plus 45 whole power 1.5. 301.86. So which is nothing but uh, 1.45 times 301.86 plus 82, 519.967. So this is the value of D naught. Now we substitute this expression here. Now we know Vr is nothing but it is 200. We know what is DO, that is 519.967. We know the value of mu g, viscosity of the gas, that is 1.8 to the power 10 power minus 5. So this is known. Now dp square is what we want to actually give that relationship to. So this will be 800. So this is the expression. If you minimize this expression and then if you substitute this in the efficiency equation, so because k is given, the value is 0.25. So if you, so this is the expression for the for psi. So efficiency is equal to 1 minus exponential minus 0.25 times 45 times psi to the power 0.5. So this will be the expression with the value of psi being 800 dp square times 200 by 18 times the entire expression. So any questions related to this? Any confusion? So here we are supposed to calculate or express the efficiency in terms of the particle diameter. Any questions? So we stop the session here. So in case if you have any questions, you're welcome to ask me, or otherwise we can conclude for today. So any questions? So shall we conclude the session? Yes. So yeah, so if you have no questions, then let's just conclude the session for the day. So thank you all for joining today's live interaction session. Our next live interaction session, that is week 7, will be on 8th of March, same 5 p.m. to 7 p.m. So I thank Professor Prasenjit Mondal sir as well as the NPTEL team for providing me this opportunity to interact with you all. So thank you once again for joining today's live interaction session, and we will meet next week. Till then, keep working on your assignment problems, and if you have doubts, feel free to email me. Thank you and have a nice weekend and a great week ahead. So yeah, thank you. Bye.